So friends, we have started our presentation. So in this presentation we are going to learn about the culture media. So it is this presentation, you can see this is introduction to culture media. So we are now we'll have to study the culture media because culture media is the most second important pillar for microbiology. Without culture media we cannot grow the fungus. So what is culture media? No, so what is culture media? The culture media is the favorable growth media of both food and environment for the microbes to grow freely. So here in this culture media, what we are going to do is that we are going to make a friendly environment, which is both food and environment, which we will be able to make for the growth of the microbes to grow freely. So now we are going to classify the culture media. Culture media has different types of culture media are there. So now we need to know how we are going to use the culture media and where to use culture media and what to use culture media. In which fungus we will use what culture media. So that we are going to see in the next slide. So this slide shows the classification of the culture media. According to the classification, culture media is broadly divided into 4 classes. So first class is the consistency. Then the next class is nutritional component. The third division is oxygen requirement. This is oxygen requirement. And the fourth one is the very important one. This is very important one. This is the functional use. Now this consistency, according to the consistency, the culture media, the consistency, how the culture media will be, or what type of what will be the consistent of the culture media. That according to that, it can be a liquid media. You can make the culture media liquid, just like solution, or you can make a semi-solid culture media, not liquid, not solid, or you can make a fully solid culture media. This solid culture media is very useful for portable. It is readily available in the market where the chemical companies make the culture media. They used to actually make it solid. So this solid culture media can be converted into aqueous and boiled. You can make it aqueous and boiled. So you can make it, you should dissolve it in water, a specific amount of water, say about 100 ml or 50 ml like that in water, and you can boil it and make it liquid, semi-liquid. Then you cool down and put it in the culture plate, and you can just, then after that you cool it down, and you can produce the fungus there. Inoculate the culture media with that fungus, or you put inoculation means putting some fungus into the culture media, and you can produce the fungus, or you can put it in the fungus, put it for growth. Another one is aqueous and non-boiled. This aqueous and non-boiled is just a simple solution. This is a liquid. As you can see, this is the liquid culture media, what we are talking about, the liquid culture media. So this is aqueous and non-boiled. You just put it in the bottle. That you have to keep in a refrigerator. For suspension, this is for suspension. Suspension means nothing. Just you can put with the host. You need not to separate the fungus properly. If you are able to separate the fungus, it is well. If you cannot, you can put with the host inside the flask. This is suspend it. You will make a suspension, suspend it, and it will be there in the flask. So this will be easiest. This is the easiest way. This aqueous non-boil, it is very easiest way. You purchase the culture media from the market. This is solid culture media. Purchase from the market, and you mix it in water, and you keep it in the refrigerator, and whenever you require, you use it. Which I have shown in my previous videos. Often you have noticed that I am taking some culture media from a bottle and from a pipette, and I am putting in the flask, and then I am putting the fungus from the host, resecting the fungus from the host and putting it in the flask. So that is that is what I was doing with this suspension culture media. So this is very easy. So now according to the nutrition component, this nutrition component, it is one is synthetic and another one is non-synthetic. Generally, these non-synthetic culture media are not available now. They are not available because they are natural. So these natural culture medias are not available now. Natural culture medias means this just the host on which the fungus is grown, or suppose for example the vegetable, or somehow it is a growth on the meat or in the fish. That culture, that host is referred as non-synthetic culture media or the natural culture media. How can you keep a fish for a long time to use a culture media? No, it is not possible. So from this, so therefore the synthetic culture media are made. Synthetic culture media is also not fully synthetic. It is also wrong. This term is also wrong. This is actually semi-synthetic culture media. What should I say? It is semi-synthetic culture media. Semi-synthetic. Why it is semi-synthetic? Why? It is because these culture medias are taking some natural ingredients and they are using it inside preserving with some preservatives. They are using these ingredients, protein. Maybe these are protein. These are protein or fat or some what should I say? Some digestive extract, or you can say blood, which we will see on the next slide. So these are taken. These are the natural ingredients. These are taken, and they are preserved by adding some preservatives and additives. So they are called synthetics, and therefore they are termed as semi-synthetic culture medias. They are termed as semi-synthetic culture medias. So they are not fully synthetic, synthetic. They are semi-synthetic culture media. Therefore, they are semi-synthetic culture medias. Now comes the next requirement, that is the oxygen requirement. Now the culture media is divided into two parts. That is the oxygen requirement. Oxygen requirement means some of the funguses and bacteria require oxygen to grow. They are called aerobic, and some does not require any oxygen to grow. So they are called non-aerobic. So this type of culture medias are sometimes used. They divide into these two properties, aerobic and non-aerobic. Generally, maximum bacteria and funguses are aerobic. Some funguses like E. coli, some funguses like E. coli and yeast. These are non-aerobic funguses. Especially this yeast. Yeast is a very non-aerobic fungus. They don't require any oxygen. So they grow in the carbon dioxide. The aerobic fungus grow in oxygen environment, O2 environment, and they require CO2 or carbon dioxide. This carbon dioxide is supplied chemically by sodium bicarbonate. They are supplied in sodium bicarbonate. So that is included in the culture media. When you will see the formulas of the culture media, I will give you the names in the next slide. We will be discussing the names of the culture media. From those names, you will get the formula. When you search in the net, you will get the formula of those culture media, and definitely from there you will understand that some of the culture medias are using the sodium bicarbonate or sodium carbonate. So therefore, they are supplying the carbon dioxide to the culture media. That you will see, that you will able to see, understand. So no problem. Now this functional use. Now this culture media is very important. This is simple media. Simple media is very simple. It consists of only agar, or it will consist of any broth. Agar is a solid. Agar is a solid. Agar is solid, and broth is liquid. So it consists very few chemical ingredients. So therefore it is considered as a simple culture media. Now this is enriched culture media. So this I was talking about. just before. It was just I was talking about the synthetic and non-synthetic natural components, some digestive extraction of blood, protein, fat. These are the natural ingredients, some of the natural ingredients which are kept in these enriched culture media. So this is required for complicated bacteria, complicated funguses, complicated bacteria, which generally develop in the human system. The funguses and the bacteria which attacks the human system, human digestive system or human system. So they require some of these natural products which are there in the host, present in the host, that is in the human system or in the vegetables or in the meat or in the fish. That environment they require. So they require fish, they require protein, they require extract of the protein, they require extract of the fish, they require the extract of the blood. So that we have to give to produce them. Now this one is very interesting, chromatic media. Now this is chromatic media. This chromatic media is very interesting. This is what chroma means color. So this media produces color. How? They interact with the bacteria or the fungus and they produce color to let us know the condition of the growth of these funguses and bacteria by the indication of the color. So if you can remember from one of my previous video, I have shown that I was using a color, I was using a agar, or rather a culture media as sodium acetate differential agar, which I will just mention after this. The sodium acetate differential agar, it was having a pH indicator, it was having an oxidation reduction indicator. So these type of indicators are added in the culture media to indicate the growth of the fungus, because a non-indicative culture media will not show you the apparent growth of the fungus. So it will be difficult to see from the flask whether the growth has started or not. If it is fungus is very slow, if it is a very slow reactive fungus, then it will be difficult to understand what the fungus is growing or not, whether the fungus is growing or not. That will be very difficult to understand. So this chromatic media is introduced. This chromatic media is introduced in the fungus. So therefore, this is used. So that we will study what type of chromatic indicators are used. So that we will study. That we will see in the next slide. Now this is the third one. This is the selective media. This is the selective media. Now the selective media is selective. This media is selective. It is selective for some few funguses, particular funguses and particular bacteria they are used. So therefore they are called selective media. This is not for all. So not unigenerous. This is not for unigenerous. This enriched media is for everything. This enriched media is for everybody, for all. But this chromatic media is also for all. This is also for all. But the selective media is only for selective organisms, selective funguses, selective bacteria. So this is why it is called selective media. It will not allow to grow any other microorganisms. It will allow particularly that microorganism which you want. Otherwise, it will not contaminate it. So this type of simple media, this enriched media, this chromatic media, they actually can contaminate your culture media with some other growths. You have to find out which you require. But the selective media will allow the only specific fungus or the specific bacteria to grow in that environment. That we will study. Now this is the third one, is sugar media. This is very simple. Three types of sugars we have, that is the lactose, sucrose and glucose. When these three are added into the culture media, it becomes a sugar media. Sugar or sweetness. That what we are seeing in the citrus fibrata, that is a sweet fungus. We added, I said that we are adding more sugar to sugar, sweet to sweet. So that is your sugar media. It consists of lactose, sucrose, agar. I said it was lactose, sucrose, agar. Less medium, so I was using a less medium. So that is lactose and sucrose. You can see this is lactose and sucrose. Here is lactose and here is sucrose. Sometimes glucose is also added. Glucose is added to one fungus. I'll just let you, for example, I can tell you that is neomycin fungus. Neomycin fungus. This neomycin is a antibiotic. It produces a neomycin antibiotic. So this fungus actually, this antibiotic is actually is available from a fungus which requires glucose to feed in. Hour to hour, you have to feed glucose to extract the neomycin antibiotic. So it should consist of glucose, and it eats up the glucose, and it releases neomycin. So this is the fungus. So therefore, this is the overall description of the culture media. So now we are in the next slide. We are going to see the types of culture media and their names. So according to the functional type, we have the simple agar. As you have seen, this is simple agar. So this is consist of only agar. This is readily available. This is peptone agar. This is also readily available. Agar and protein. Now protein is added here. Protein can be beef extract. Protein can be beef extract. This is also available in the chemical market, and it can be fish extract. It can be bile extract. These are all available in chemical market as culture medias. Reagents, it is readily available as reagents. So all you have to keep in refrigerator, because this beef extract, this fish extract, this bile extract, bile salt, another one is there is called bile salt. These things are readily available, but they should be kept in refrigerator, otherwise they are spoiled. They will get spoiled in high temperatures. They will get spoiled. They need to be refrigerated. You have to keep a refrigerator in the culture media, in the lab. I will be showing that thing also in my future videos. So you need to keep a bile salt. You need to keep a refrigerator. So now this enriched culture media, as I have told, this is consists of blood agar. So this is a special type of agar. This is available. This is readily available as a reagent. This is blood agar. This chocolate agar. This is also readily available. Now this agar with vitamins and amino acid and buffer mix. So this agar is a very special type of agar. This is not used readily for culture media, or not readily available, not readily used in microbiological analysis or microbiological fungal growth. This is used, and the second one, this is Young's balanced solution. These two are specifically used in bioreactor. This is specifically used in bioreactor. Bioreactor is a specially designed equipment. I will be showing you it in the last video of this session. This is bioreactor. So this I will also discuss. And bioreactor is a specially designed equipment where you can use, it is a very sophisticated instrument where you can use this type of culture medias, this vitamin, amino acid and this Young's balanced solution you can use to reduce bacteria over there. It is a controlled environment. It is a very controlled environment. It is electronically controlled environment. You have pH indications, you have dissolved oxygen indication, you have lots of temperature indication, you have all lots of things in there to monitor. So there these type of agar mediums are used. So this is enhanced, this is a very enhanced and advanced culture media. Enhanced, or you can say not only enhanced, you can say advanced culture media. Advanced culture media. Now the third one is selective, which I was talking about. So this is Trigalaski agar, this is a clade agar, and this one is sodium acetate differential agar. This I was talking about. This is also a chromatic agar. This is a chromatic agar, and the third one is Ramback agar. This is also a chromatic agar. Now, especially this chromatic agar, if I talk about, this is consists of a chemical compound called ONPG. Another one is XCal. These are readily available. You don't worry. This is readily available. Readily available in chemical markets. So that doesn't worry. You need not to worry about. And formulas are also available. If you type in the Google search, you can get all these agars, name of these agars. You will get all these name of the agars. No worry. This is it's all you will get. This digalactic agar is selective agar you will get. This clade agar you will get. This sodium selective agar get. Rambach agar you get. ONPG also you will able to get. Information about ONPG that also you will able to get. XGAL you will get. This lactose sucrose agar definitely you will get. This is a sugar agar, so definitely you will get. This agar with carbohydrates. Another one agar with carbohydrates. This is the glucose is there. Different types of carbohydrates are used. So this type of agars, these are sugar agars. These are all available in internet. Formulas are available in internet. You can bring the formulas and you can just download the formulas and you can just make in your lab. It will be economical. You just process the raw materials and you can just make it in your lab. It does not mean that you have to make a big amount. You can make a small amount. You can make about 100 ml. For 100 ml, you can mix them in 100 ml of water. Few milligrams, you can have 100 milligrams of agar. So that you will do. So this is all about culture media. So now we are finishing up the video and thank you very much.